So just for the people joining us, so we have kindly agreed that we can record this presentation as well. If you place your questions in the chat or Q&A, I will monitor this. And I will, towards the end of the presentation, just try to go through these questions. If people prefer, just put in the chat or the Q&A that you would like to ask us in person. If time allows, we will go through that. So we are quite flexible. And we will just otherwise do this in a sort of normal format. We be happy to try to, we will have time to go through all the questions. So we will talk for about 45 minutes or so, but it's going to be very, very interesting. So I will not hurry him along. I've been looking forward to this immensely. Because Huang, who's now been at the Institute for Citizen Biology in Seattle for some sort of 12 years, 10 plus years, has really done the most pivotal work in this area, trying to give a qualitative scaffold and interpretation to Waddington's metaphorical landscape. And his work has been hugely influential for lots of people, including my own group, because it convinces in terms of originality and clarity of thought. And I'm immensely looking forward to Sui's presentation. Now with people coming in, we always allow for a couple of stragglers, but I think in the interest of time, and I can't keep my children quiet for much, much longer because they're also in the audience here, I will start and invite Sui to take to the virtual stage. I look very much forward to listening to what he has to say about cell state transitions, attractors, instabilities, and implications for cancer treatment. Sui, please.
Thank you very much, Michael, for this very kind introduction. So it's very exciting for me to present to this audience, which I haven't seen. And so I assume that it's very diverse, and I hope to also reach especially experimentalists. I think that a challenge for them to appreciate some theoretical concepts. So everything would be graphical, and I hope that I can convey some key concepts. As you will see, it is shown here. So as Mike mentioned, this famous epigenetic landscape metaphor, and then we'll move to more theoretical basis of that, and then I'll show some data related to cancer treatment. But let's start with a bigger picture view, and that is a prologue about the evolution of the concept of genotype and phenotype, which I think is underappreciated. It's a very complicated, winded history. So one examples are humans who adapted to high altitude life, hypoxia, very thin air, like 4,000 meters, and how they do that. If you look at here, in the Indians or the people in Tibet, they use different strategies, right? In the Andes, you have people have a high hematocrit, a lot of erythroid, red blood cells, more than normal, but in the oxygen saturation is almost normal. Actually, it's normal. In Tibet, you have normal hematocrit or red blood cells, but much lower oxygen saturation, but they compensate that with increased blood circulation, whereas the one in the Andes have slightly lower blood circulation. So people have been interested in that, and of course, one thing to do is look at genes, and they found SNPs, so polymorphism in the genome. They don't overlap at all, except for one. But the point I want to make is that we have to look beyond genes. And if you look at how humans adapt to high altitude, there are two time scales. Right? On the left is when you go for a few weeks in the mountains, Colorado, or in Tibet, trekking. For weeks, you make the same changes. You create more red blood cells, you increase ventilation, and so on. But then on the right, you have the evolution over years. So you have acclimatization, which depends on physiological plasticity. And on the right here, of course, one would assume that there are mutation and selection for the phenotype that is adaptive to hypoxia. And these two are related, and very often we forget that. In fact, some people say that we can only evolve this high altitude phenotype because of this concept of the evolutionary adaptation tracks physiological adaptation. So individuals can adapt reversibly, and because of this plasticity, that's exploited then by evolution. Now, how we evolved that plasticity in the first place is a totally different question, and we're not going to talk about that. And so people have been studying that, and if you look at these two populations and you measure ventilation volume, you can see dramatic differences, not so much with respect to the average, so every dog is a person, but in Tibet, you see this widespread between, so male and females, different colors, it doesn't matter, but one lesson that we can learn is when we measure this type of things, it's important to be aware of the population structure, so we measure the variance and higher moments, and then we measure a population structure as a property, and for that, we need to measure individuals. At the resolution of individuals, which is easy here with humans, but in biology, in biochemistry, we have for decades just measured entire cell populations as one amorphous mass and lost a lot of information in doing so. Another example is the evolution of bipedalism, and this is an example that is also mentioned by Marie West Eberhard, one of the proponents of plasticity in evolution. So most people would think, oh, well, you gain mutations and you select, and then eventually we have all the mutations that is needed to have this bipedal gait. But it might be not as simple, and that's most dramatically shown in this example here of a goat which was born without 4 legs, but it could be, it could be accidental or genetic or, a brutal experiment. In any case, they learned to walk with 2 legs, and in the 1940s, people have dissected the anatomy of after, when the goat dies in high age. You can see on the left here the normal anatomy, and on the right is the anatomical change of the pelvis structure, and it really looks like that of a human being, much more closer. So now you can imagine that if these goats were born in an environment where everyone gets, chopped the 4 legs, or they have to stand up to eat a high-growing tree fruit, then they would all have this type of anatomy, and we wouldn't ask whether there are mutations that confer that phenotype. So that complicates things, but it all started with Mendel, very simple experiments. This is high school biology, Mendel rule of inheritance. You have 2 peas, one is smooth, one is rough, and if you cross them, you get this 3-to-1 because rough is dominant. People have been thinking about how this could happen, and it was a mystery because people didn't distinguish between phenotype and genotype until Wilhelm Johannsen in 1906 came up with a brilliant experiment. So he looked at bean sizes, and then he took different, what they call pure lines from long back crossing, and he made this histogram. This is a physical histogram, and then he got the idea that one has to distinguish between phenotype and genotype. So every histogram here is a genotype. It's a pure line, it's a clone. And within every clone, you have variability, and that's phenotype variability. And here in the bottom is when you have a mixture of different genotypes. So that was the beginning of the distinction between genotype and phenotype, and it took actually quite a while for people to accept that. But Schrödinger got it actually independently, I guess. So in 1944, as you all know, What is Life was his book where he proposed existence of DNA and so on. But a less appreciated fact is this figure that he painted on in figure 7 when he asked whether, when he looked at the height of this wheat, whether this variability is genetic or not. And so he proposed an experiment where you take, again, a purebred crop, and then you would pick out one size by sorting for a particular size and regrow again. So he had already the notion that there is phenotypic variability that needs to be differentiated from genetic variability. And I will come back to that later, to this experiment. But now, some more basics about cell states. What is a state space and attractors? So these are the early concepts, and then later on I will move to a more formal description and then applications to cancer biology. So we're interested in cell state transitions. These are differentiation, from one progenitor state to a differentiated state, or sometimes you have this branching development where one multipotent cell can generate two lineages. These two phenomena turned out to be very closely related. They're actually the same, as you will see later. So if you take a cell, and I think most of you appreciate that we have stem cells, and then the stem cells generate all kinds of cell types in the metazoan body. And we can now measure the gene expression profile of these individual cells as a state. So these are the gene expression levels of M genes, could be in the 10,000s. And that's the way we display them. This is just a self-organizing map where every position in this map is the same gene. And you minimize the differences of gene behaviors across the entire set. And so you see visually characteristic gene expression profiles that are very robust. These are just two individuals for the same cell type. So how do you get these robust profiles, these vectors, every time you develop? Obviously, the genes are not independent entities. They're connected by a gene regulatory network that influence each other. And this influence constrains the configurations to which every gene can have a gene expression level. So a messenger RNA level that you can measure. And you can write these as just a simple ODE, where the expression level change depends on the state of the entire vector. So that's for those that are interested in this formalism. Very straightforward. Now, reality is different in development. The stem cell just doesn't create all these cell types at once, but it undergoes a development which has always this binary branching to populate all these possible cell types. And that is why Waddington thought to capture that with this valley here. And the two properties that he found was that you have discreteness of cell phenotypes, the cell type that you observe of blood cells, liver cells, and so on. And typically you have binary branching. And he also realized that intermediates are rare and not stable. So that's how this metaphor came up. And now let's look at it in more closely. So if these values are individual states, it would be a cell type, like here. And another one would be less differentiated. And development would be for these cells to roll down here, or to move from this gene expression profile to this one here. But it cannot do so freely because of the underlying constraints from the gene regulatory networks, which dictates, like, for example, if this gene is up, then its target must go down because it inhibits it. So we have a complicated problem to solve, and that is that you cannot just ad libitum gain any gene expression configuration. So it's highly constrained, coordinated process to achieve a particular configuration. And if you differentiate, you have to go from one stable state to another. And that's the challenge we'd like to explain. Waddington also coined this term create the necessary path. So this gene regulatory network, of which he didn't know anything, would constrain the path through which a cell changes from one phenotype to another. Okay, now I'd like to, in a very condensed manner, mostly for the experimentalist, give you an idea of how we get from network to a landscape. So we have this landscape, and what we need to know that nowadays we explain development using this type of diagram that you see everywhere, these pathways and wiring diagrams. A simple version would be, for example, typically this linear cascade where you have an input, a cause, and an effect. But of course, this is not as simple, and people early on realize that you have a network with a lot of feedback loops. So another way then to take care of that is not to just rely on this cascade of cause-effect relationships, but to map this entire network here in a state space in which every axis now is the expression level of a gene. So you have A, B, C for these three genes. And these are the displays you see in papers when you measure them as a profile. B is high here, as you can see, and A is very low on this axis. Okay, so every position now is a gene expression profile. And the whole thing, the whole network here maps into one point at a given time. And as it changes, gene expression profile, position changes, and you move to another state. So that's very simple, basic idea, but still this arrow here is a trajectory, whereas this arrow here is just kind of a, back of the envelope arrow to capture some causal relationship. So, and you can do that for high dimensional systems. So you would move from one transcriptional state to another. Now, one can also show that there are many paths that can go to the same state. And often the system will settle down into a stable state when no change is possible anymore because all these interactions are satisfied. So that's the idea of what is called the attractor state that attracts nearby trajectories because it doesn't violate any of these rules of interaction between the genes. But now we like to move another level of abstraction. And that is, how do you get this notion of a landscape, which is very abstract? And you need that because you want to relate different points. So we take this space, and this is just pure for display. So of course, if you reduce the dimensionality, you lose a lot of information, but still you can capture some neighborhood relationships where nearby points are similar. And then this frees up the third dimension to plot for every state in the system, some value, some potential value. And then the idea is that every point on this landscape will correspond to a particular state and would be driven by these interactions to seek points that are kind of lower potential value, so to speak, because it satisfies all these interactions. So the main point here is that then you have trajectories that kind of go down to the lowest point. It's a very dangerous metaphor, but it's helpful in many ways. That's why we call it a quasi-potential landscape and not an energy landscape. But the point is that now you have multiple attractor states, local minima. And with a landscape, you have a tool to relate the relative stability of these local minima. And that's the whole point, which you don't have here in the classical dynamical systems where stable states are just analyzed separately. So the idea that we introduce is the relative stability of multiple attractors or any 2 points. And we will summarize that in very simple language in these papers here. So now, this led to the idea that Waddington's landscape corresponds to this quasi-potential landscape, which is informed by the underlying gene regulatory networks. And this was the old idea first, as I mentioned, proposed by Waddington in a metaphoric way. Delbruck introduced bistability with 2 attractors, and then Giacomo Monod discovered gene regulatory networks, and Kaufman extended that to big networks. So, so we have the correspondence between these two, and we have a state space and some sort of relative stability for every point. So that's the basic idea. Now coming back to this here, this experiment of Johansen and Schrödinger. So we started to apply that to cells and to see whether the fact that you have this distribution here might reflect some of these dynamics. So the idea is you have progenitor cells and they're in an attractor state and they can go into a differentiated state. And they are stable in that the majority of cells are in the center, and then these outliers are those in the basin on the border. So we took a cell line. It's important to be clonal so that this diversity is not due to genetic diversity. And we also did careful measurement to make sure that this variance here is not due to measurement noise. The measurement noise is like 1/10 to 100th of this spread. And then using FACS sorting, you can now sort these fractions of cell population and measure the transcriptome. And when we did, first thing we realized is they're different, not surprising, but they're different with respect to many genes. So we have a high dimensional dispersion, not just with respect to this SCAR1, which is just one thing we can measure and sort. So that's a cell surface marker. And then if you leave these cells in culture over time, they would reconstitute the population in the center. So sorry, that, okay, good. So you have a convergence of high-dimensional trajectories, which is very strongly indicative of an attractor state here with respect to many variables. So we have this population heterogeneity, which is bounded by an attractor basin, and they will spontaneously relax to the center. Now, a little bit of a technology. So the measurement I've shown you is using a traditional microarray that we have been using for decades now. So you take a dish and you essentially measure only one vector, which is your transcriptome. And then you apply a perturbation, you measure another vector, and then you try to figure out to learn something from this change of this state vector. Now, I think most of you have heard we have single cell resolution measurement where you can take individual cells, for example, along X1, you can sort them or just randomly. And so you measure a matrix where every column here is a cell and every row is a gene. And then you gain additional information from the population distribution with respect to this high dimensional space. And you can again perform a perturbation. And now you change not only gene expression profiles, but also cell population structure. And that's something that we want to learn about what happens. So how does this matrix change upon perturbations? It must somehow reflect this network here. So some theory now a little bit, because I just showed to you, the existence of this landscape. So the two questions that we can ask, and the first is, what does really this vertical dimension, this elevation means? It's kind of a potential, but what is it exactly? That's one question. The other is, how do you go from one attractor state to another? And I mentioned the notion of relative stability, and that's why it is important here. So if you have a vector field that describes the dynamics of these genes vector, typically you can have a noisy system, and that's important because without noise, you are always in the attractor state, and the individual attractor states don't know of each other, right? So you've talked about transitions, it can only happen in a stochastic system with stochastic noise. And one goal that we like to achieve was to see whether this landscape in some way reflects the architecture of the network, which is essentially F, which we don't know typically, right? So it would be easy if you can write it as a gradient of X, as gradient field, but that's typically not the case in this high dimension. But what you can do is split this in two components, and there are many ways to do that, and many people have proposed many ways. One way is when you have these two components perpendicular, where you have a gradient field, and then something else, G, which shall not bother us at the moment. And one can then show that this potential correspond to the Friedling-Wenzel potential function, quasi potential in their large deviation theory, if this decomposition is perpendicular. And what it means is that you have a probability going from A to B, which is exactly related to this Arrhenius-like relationship, so that the potential difference tells us about the probability given this noise for a state here to get out of this attractor state. But the good thing about this formulation here is that you can just, in principle, numerically compute a landscape without stochastic simulations. And therefore, we can have, if we know this function here, we can have a notion of a landscape that captures the dynamics quite well if G, this component here, is small, which turned out to be the case in most cases, and many people have studied that. So in living systems, it just makes sense to have this decomposition. And it's probably not generally true. So there are many theories about that. Some people try to use just without this mechanistic notion, and I found it elegant to just define some potential landscape and use differential geometry and constraints there to also try to understand cell behavior. But the main point is that this quasi-potential landscape is a structure that manifests stochasticity in this network in some deterministic way, because we have this unique mapping in the limit of zero noise, but you need noise to have the notion of hopping between attractors. So that's the idea. And the second question is, how do you model now, given that idea, going from one state to another? And I mentioned that we are interested in these transitions and the different ways, right? One is you have these rare spontaneous events, but the question is, how does a signal trigger such a transition? So a very old idea is you have these rare random transitions, and the signal is nothing more than a selection. So for example, a state B would express a receptor for this signal, and the cell would then proliferate, would grow. And in fact, there's a lot of evidence that this component plays a role. A lot of differentiation factors are actually growth factors for this destination state. The second one is somehow the signal pushes the cells up some least action path. And then when you achieve this point here, then you are free to roll down to this new state. That can also be possible, but given the high dimensionality, it would be a very complicated signal, and we know signal transduction is very complicated. It involves many genes. But then what we thought a very simple way would be for the signal to change the landscape, because as you know now that the landscape is really, it reflects the parameters of interactions, right? Because it's essentially the system here. So the signal will make it flat, almost like a catalysis, and then you have no energy barrier anymore, and gene expression noise would drive cells into this new state. So we kind of favored that as one possibility, and probably in reality of all three that play a role. So in this case, it's this probability to go from A actually to this top here, which matters. This would be slow. This, as I mentioned, would require a lot of genes. And here, the advantage of that is really one single parameter change, and then you might have this change of the landscape. So that brings me to the idea of self-decision decision or transition as a critical transition. So, and we look now at this branching. I think most of you have heard of critical state transitions in ecosystem, where you move from A to B to a new ecological state or climate. And here in this simple transition is signature of eminent transitions. This critical state is that is early warning signals, larger fluctuations, associated slower relaxations of the attractor state, and long-range correlations in time or in space, depending on your system. Now, what we have is a much more complicated system, a high-dimensional network, and a complicated landscape. And when we destabilize a state, so the condition to get out of this attractor state is really a destabilization, then you lose control and cells might go into the state that you want it to go, given the signal. So a particular cell could be here, and you have this vanishing of the attractor state. That would be the desired state, but because of the complexity of the landscape, it's hard to steer everything into one state. So you might have cells that spill over to totally alternative state, opposite to that you want in some way. And we call this the rebellious cells. So essentially, if you want to go to state 2 in a complex landscape, you might accidentally get these aberrant cells that go in the other direction. And what we have now experimentally is we can only have discrete time points. So we cannot do this type of correlation in time. We have a high dimensional system, but these are the disadvantages. But what we have that this ecosystem people don't have is we have a huge ensemble of systems. We have many, many individual cells that form a population or a statistical ensemble of systems. And these are these columns here. And so we like to take advantage of that. And what we propose is that instead of this critical slowing down, we'll have an increase in cell variability, which is intuitively obvious. And as equivalent to this long-range correlation, we will have a coordination of the genes, so which are the rows here. So that is what I like to show you very quickly. So if you have just a state vector of one of the average system, And this is a classical bifurcation, for example, pitchfork bifurcation, which is just one example. The idea is that you have to destabilize your attractor state, so you lose stability, and you have these increasing fluctuations that you capture in one single time point. That's obvious that the cell-to-cell variability would increase, but what is less obvious are the correlations between the genes. So you have here, we also want to show that the genes here correlate, and so you use a discrete system, assume a hyperbolic attractors, all eigenvalues here are negative, and that there's a quasi-equilibrium approach to this bifurcation. So this is relatively slow, and then you can write down, since we don't know F, so the exact structure of this network, what we know is that one eigenvalue in this discrete system would go to 1, and then essentially ask what happens to the rest. So we can write this equation now. This is Jacobian now as a expression of the eigenvalues, and to capture now this variability here between the cells. So we can add Gaussian noise or even non-Gaussian noise, it doesn't matter. And you can rewrite this as these statistical expressions, and then show that if one of these eigenvalues goes to 1, this correlation would also dramatically increase as you approach this critical point here, and the states, the correlation between the states decrease. That's more obvious, and therefore this index here would increase as you approach the critical state. And so the general idea is that because you have a whole population of cells, you have an ensemble of high-dimensional systems, and if you can measure individual states, these high-dimensional vectors, then that would reveal the change of the landscape and the destabilization. And so you can see when there's an impending bifurcation. So 2 predictions is this increase of this index, and then which is due to gene correlations increasing and cell correlations decreasing, and then the appearance of cells that totally move in the wrong direction, which are our rebellious cells. One way to show this, to intuitively understand that, is shown here. So what changes if you move from an attractor state to a transition where it flattens? In the attractor state, the cell correlation are very high. So these are, every dot is a gene, and you just compare a pair of cells. 2 cells are very highly correlated with respect to their gene vectors, right? Because they are essentially identical. And then cells become different in transition, and so the correlation decreases or the cell-cell variability increases. Now, if we look at now the row, so gene correlation, it's less intuitive, but in the attractor state, if there's no noise, all the genes should be identical in 2 cells. Sorry, all the cells should be identical with respect to 2 genes, because that's defined by this fixed point here, except for noise, which is symmetrical, and therefore you should have no correlation. But now in transition, you have, for example, a saddle point, cells aligned along these trajectories, and you have a reduction of the dimensionality of the manifold where all the cells are, and so that increases the correlation between the genes. So that's the basic idea, and so we expect that this index increases. Can we see that? And this comes now to the experimental part. So we take, again, our system, these EML cells, which can differentiate into erythroid and myeloid, or red blood cells and white blood cells. EPO, like the people in the high altitude, if you give EPO, they become red blood cell. If you give IL-3 and GM-CSF, they become myeloid. So it's a bipotent progenitor cell. And then we would like to see whether these 2 predictions can be observed. So this increase of the index and appearance of cells that go in the wrong direction. So this was the early days of single cell. We did PCR, which turned out to be much better because much more sensitive. And you can see here, this is day 0, the entire population. Day 1, they moved. And the colors represent now the label of the treatment and not any clustering algorithm. So cells treated with EPO are red, cells treated with GM-CSF are blue. And then we also tried the combination treatment to force the cells to not know where to go. So at day 3, you can see a further spreading of the cells, and the purple cells are in the middle, the cannot side. Day 6, now you have the red, a nice separation between the red cells and the blue treated cells. So what you can see here, these are the destination attractors. You can see also at day 3, characteristically, you have blue cells in the region of red and red cells in the region of blue. So these cells are moving in the wrong direction, and we can analyze them. It turns out that they later die because they don't receive enough support from released factors. But what is interesting is the combination treatment. They cannot decide. They stay longer in this intermediate state, and later on, for some reason of asymmetries we don't understand, they move toward the myeloid state here. And then we can compute this critical index. So just to show you the raw data, this is just a correlation matrix. So cell by cell, you can see highly correlated, and it gets lost, except for some pockets of cells that differentiate early. And then you increase the correlation again. If you look at gene-gene correlations, really the correlation initially, then day 3 of high correlation, also anti-correlations. In this, also the genes, the transcription factors that are important, involved in the transition, and then it decreases again. So if you compute that, sorry, you get a sharp peak of this index around day 3. So that's a signature of a critical transition that we like to use. Now, we like to look closer at these rebellious cells. So these are the cells that move in the wrong direction. And this is an interesting experiment. It's totally counterintuitive. So we have, again, this one-dimensional variance of SCAR1. So that's a surface protein expression. If we treat with EPO, you get this erythroid. If we treat with GM-CSF, you get myeloid. And that manifests in the appearance of the low SCAR1 population, because SCAR1 is a marker of the progenitor cell. So cell lose SCAR1 as expected, but not every cell responds. Here too, you see the appearance of the SCAR1 low cells, and again, not every cell responds. So you suddenly see a bimodal distribution. If you look at the nature of the cells in these populations, so first, untreated, you see expression of stemness genes like c-KIT, c-MYB, and SCAR1. These are the cells that responded as expected. Here you see erythroid genes, EPO receptor, hemoglobin, GATA1, the classical marker of red blood cells. And here we see these myeloid genes like PU1, CD11B, and so on. So that's expected. These are the genes that responded. But what about the one that appeared to not have responded, at least with respect to SCAR1? You can see that these cells here that didn't respond to GM-CSF, if you look only at this marker here, actually have gene expression profile that are almost identical to cells that were treated with EPO. So they move in the wrong direction. Similarly, these cells here that appear to not have responded to EPO, that's B population, they have expression of these myeloid genes, except they keep some stemness genes, but they clearly have signature of these white blood cells. So the non-responders appear to have responded just by going in the wrong direction. And we can even look at this in more detail. So this is an old diagram. It turns out that these two transcription factors determine in which direction you go. They are reciprocally activating each other. And PU1 turns out to be correlated to SCAR1. So SCAR1 is a good marker for PU1 level. And if we saw these two tail populations and now functionally analyze what happens, it turns out that the SCAR1 low population, this left tail, are much more prone to differentiate into erythroid if you give EPO, and much less prone to turn into myeloid cells if you give GM-CSF. And the opposite is true for the SCAR high populations, meaning that these cells are already fated or primed to differentiate in these two directions. But if you do nothing, as I've shown earlier, if you just sort this out, they will return to the center or to the other side. So cells in the tail fraction of any distribution of this clonal population are primed towards distinct fates. And so we took advantage of that and do single cell analysis now in these populations. And what you can see here, this gets a bit complicated, but if you treat all these three populations with these two signals, you can see here again the attractor position for the future myeloid cells on day 3, that is this intermediate state, and here the erythroid state. You can see that the one that went wrongly in the wrong direction are the light blue one. So they are the one that came from the tail population that we know now that they are primed to become erythroid. Whereas the red ones that went into this blue region transiently are the dark red one. So these are the ones that were treated with EPO but become myeloid because they came from the SCAR1 high population. So that fits with the fact that we have this intrinsic bias already in this heterogeneous population. These are the other rebellious cells here. So the initial non-genetic variability is not just purely random, but it manifests some internal constraints in this high-dimensional space, and it affects the responsiveness to signals and future states. So now I'd like to apply that to cancer in the last 5 minutes or so. You probably know that there has been a big change in cancer therapy from chemotherapy to target selective drugs, where you target a particular pathway. But the success has not been overwhelming. This is a summary from this paper here by Fojo, where you can see that the majority of FDA-approved drugs in the last decades, they didn't extend life by more than 6 months typically. But something is not right, and that's what a lot of work is going on now. Why are these targeted therapy, which have a pathway rationale, why don't they work? And that's related to a broader phenomenon that I like to present here, and that is, why do things backfire in real life? For example, the war on terrorism. If we just bomb them, we know we empower them and promote recruitment. That's well appreciated. If you try a coup d'etat, often if the dictator survives, and it strengthens, and embargoes typically also, if it doesn't kill the dictator, it will strengthen regimes, and attacking your political opponents will fire up their base. So a lot of things in complex systems, there's so much internal energy, you cannot just, it's not a zero-sum game, and that we think is also what happens in cancer treatment. So in these complex adaptive systems, there's no partial response. You cannot come back and finish off, because after the first not fully successful strike, the system is stronger. And so any intervention is a double-edged sword. And so we'll try to show that in cancer. I've shown you, this is a summary of the data I've shown, and we have shown this now for this type of bifurcating development. Essentially, you have a cell, and because of this destabilization and access to 2 new attractors, I think that's what nature utilizes to differentiate a progenitor cell into 2 cell type. And you can have rebellious cells in each direction, but in cancer treatment, something similar. So we try to push cells into this apoptotic state or into some quiescent state. And if we don't succeed 100%, you get cells that are essentially rejuvenated. They're like stem cells. So, and that's important now for resistance and why cancer comes back. So the whole idea is that every new phenotype of a cell is due to a mutation, whereas now we have to deal with a lot of plasticity of a phenotype given the same genome in the absence of mutations. So the left view is compatible with Darwinism that people use to understand evolution of cancer cells in the body, whereas plasticity would complicate the entire picture. So just to recap, in the current view, why you have resistance is that you have a mutation that creates a priori a resistant cells. You create then with the therapy the green cells, which are susceptible to cell death following chemotherapy, and then you select for the resistant cells. Whereas in the new hypothesis, we have this plasticity. And what you should also know is that no therapy kills all the cells because of the variance of relative resistance of individual cells. So this is a kill curve, is essentially the cumulative of this histogram of responsiveness to any drug. So even at 90%, at a high dose, we kill 90%, still always cells that survive. It's very hard to kill 100% of the cells. And what happens with the non-killed cells? We've never studied them. So here is what we think happens. Your goal is to push these sensitive cells into the valley of death, the apoptotic state. But because therapy perturbs the entire system, you not only get dead cells, but also cells that are pushed in the opposite direction and become rejuvenated, or they acquire stem-like features. And so the system is destabilized and cells spill over into other nearby accessible states that you cannot control. And of course, you can also select for these cells here. So this idea does not exclude traditional Darwinian selection, even if there are no mutations involved. So here you have on the left mutation and selection, and here you have an induction of a new state by the actual selection pressure, the drug, and then you can have selection of a non-genetic state. So this is a classical survival of the fittest on the left. And this response here is what we summarize with Nietzsche, that is, what doesn't kill me makes me stronger. And I think it applies to a lot of complex systems. And so you cannot not respond. The non-responders actually respond in other ways, and they might, some of them at least, might become stronger, and then therapy backfires. So that tells us that we need to consider both sides for evolution. You have this intra-individual adaptation. So the first experiment we did for that was bulk analysis. We measure MDR1, which is a surface protein that also confers resistance, well characterized. And you can see if you treat these leukemic cells in order to kill them, you get actually survivors. So this is sorted for the survivors, and you see within a few days the appearance of a new population B. And it turns out what is important is that this MDR confers resistance, and we can now analyze the subpopulation. So what is important is to distinguish between A and A prime. Again, A prime looks like the non-responders, but they have changed because they were, for a long time in the presence of selection pressure of this signal that changes the gene regulatory network. And so if you analyze them, these again with transcriptome, you can see that A prime looks quite similar to A, but it's not identical, and B, as expected, is totally different. But you see many genes change actually in this apparently non-responding population that is different from the never treated population. So those are not the same. And if you can look at the genes involved, and you see typically wind signal upregulated and a lot of stress genes being upregulated, resistance, detoxification genes of biological toxins, and stem-like genes. So again, you cannot not respond. The cells that don't die, they change the internal state. So then you can explain now this model actually with the pathways. I'm not going to that, but for those that understand those diagrams, this mutual inhibition between stemness and this apoptosis pathway, you can write down the equations and show that you can get this bifurcation. And so we did the gene expression profiling of the cells that survive, and using this standard now single-cell transcriptomics, you can see here the blue are the untreated cells, and after 24 hours, if you just sort for the non-surviving cells, they really shift the expression profile. So this is just a dimension reduction in 2 dimensions using t-SNE, and they further change after another 24 hours. You can do cluster analysis and show the different distinct clusters. Even among the untreated cells, you can dissect into many, many different subclusters. And then now we can look at genes and what are these cell types. So it looks like some cell types looks rather like stem-like cells. And then also here again, the expression of many nasty oncogenic genes that you don't want to have, that they get induced by the treatment. In a small population of cells, I have to say that not every cell have all of these. So now quickly in vitro, is that true? So this collaboration with my former mentee, Deepak Panigrahi in Boston. Here's this mouse model where you treat a very aggressive tumor of the lung. Untreated, you inject 1 million cells. If you treat with cisplatin, you reduce the growth. But now if you inject only 10,000 cells, which normally give rise to very small dormant tumors, like here, you can see, barely see, but they are there. And this is a different axis. Now you treat it with the exact same chemo, same dose. You actually awaken in some of the animals, the tumor, they suddenly started to grow. So it's just the opposite from what you expect. And typically people don't do this type of experiments because typically you grow as aggressive as possible a tumor in order to show that your drugs is worth anything. That's what pharma industry does. But if you treat slow growing tumor, you can actually stimulate their outgrowth. And we tried that in many tumors, like in the breast cancer. Fast growing tumor, you treat with, this is a estrogen positive tumor, so you treat with tamoxifen, and then you see reduction as expected, but in a very slow growing tumor, the same treatment with this anti-estrogen would stimulate the tumor. And the same here with a lymphoma, slow growing tumor, here without treatment, and treatment would induce the growth. So, and finally, we try to see whether this index is any use in the clinic by telling you whether a tumor is rather in this stable state here or on the way to destabilize and become, more stem-like and metastasize. So this is from some early data collaboration with Joseph again and Anna Verauto in Helsinki. We look at 25 ovarian tumors. These are individual cells in tumors, but we focus only on the epithelial cells, which is in this red frame here. And for every of the patient, we have this matrix here, and so allows us to compute the index. And so we do so, and we also know the clinic of what happens with these tumors. So on the top, you see now, so you arrange, these are clinicians like to do, you arrange every patient in the order of the index, so these so-called waterfall plots. And then you color them according to some phenotype. And you can see here, on this end here are the ones that are less stable because they have a higher index. The majority of them will metastasize, whereas some that are remaining local tend to have a lower index here. And you look at progression after the chemotherapy, similar picture here. The one that progressed tend to have a high index, whereas the one that have a more favorable course, at least in this observation time frame, they have a much lower index that you can compute now from single cell analysis. So that's probably the idea of capturing the inherent stability of a cell population, of the state of a cell, without looking for particular progression markers that most people do. So progression might not necessarily be indicated by expression of some marker molecules that people now chasing after, but it could be in the patterns of a cell population. And I think that's what we should try to learn to do now that we have single cell analysis. So I think I'll stop here. Let's summarize. Take-home message. So the cell phases are stable attractor states. Switching between them, we propose it can be explained by bifurcations, where the state gets destabilized and opens up to new stable states, and that's why you have this branching development. You go through a state of instability where you lose controls and cells can move in undesired states. And we see that also in reprogramming, when we reprogram stem cells to a particular state. So it reminds me of this hydra here, where you have to kill all the heads. If you don't, then you will grow more heads. And that's important to appreciate when we design new therapies. So it's what doesn't kill makes me stronger. So I think I stop here, and thank you for your attention. I'd like to thank the people here, mostly these people here. You see Amy and Angela, they started with drug resistance, they moved on now in the leading positions. Mitra did the single cell analysis in the early days with PCR. A lot of theory come from Joseph Zhu and Alex Kupin, and have fantastic collaborations and inspirations from all these theoreticians here. And Deepak did the mouse experiment. Thank you.